Good morning, everyone. I know the residents and fellows will be trickling in from their didactics, but I wanted to go ahead and get started. And so, it's my distinct pleasure to introduce you all to Dr. Bartosz Grzywacz. You all probably know him. He's been here nearly 6 years. But it's really fun to see our own faculty do grand rounds, and I really appreciate Bartosz's opportunity to rise to the challenge. Sometimes January is a month where we don't get as many external speakers, and Amy just put a call out to faculty at large saying, do you want to present? And so without my nudge, without the mentoring committee's nudge, Bartosz said, you know what, I want to do this, and volunteered to do it. And that's really remarkable. So Bartosz, as you know, is from Poland. He went to medical school at Wrocław Medical School. And after medical school, he trained in internal medicine and had specialty training in transplantation. He got really interested in research while he was there, and so he wanted to have another opportunity to learn more about immune therapy. So he came to the University of Minnesota in 2005-09 to work with Mike Verneris, who used to be here. And at that time, he did a lot of really nice work looking at NK cells and kind of figuring out where they derive from and maybe how they could differentiate from myeloid progenitors. He subsequently trained in pathology at the University of Wisconsin and then did hematopathology training at the Cleveland Clinic, where he got a lot of expertise in T-cell lymphomas. So he's been on the faculty 6 years, and today he's going to tell you a little about NK cells. So he had a very basic background in NK cell biology in the past with Mike Verneris, but now he's going to tell you a little bit about how can we use and think about NK cells in disease. So in hematopathology, how do you better define NK cells? How do they present in disease? And then in immunotherapy, if you're using NK cell therapy for patients with myeloid or other neoplasms, how do those NK cells disseminate? So without much further ado, Bartosz, thank you so much, and I look forward to hearing your presentation.
Thank you very much for the very kind introduction and for the opportunity to present. And so we will be talking about NK cells in clinical and research studies, and also in hematopathology and immunotherapy. And short outline, we will talk about first NK cell differentiation, the research I did on that topic. We will discuss a case of unusual lymphoid precursors that has something to do with that first part. We will talk about NK cell immunotherapy and aggressive NK cell lymphoma, which is EBV positive unless it's negative. And so, short introduction about natural killer cells. Natural killer cells are a little bit like myself. They specialize in negative thinking, so they don't recognize, like T cells recognize non-self. They don't recognize foreign. What they're sensitive to is the lack of self, and that's the difference. So it's a little bit easier to define NK cells, but what they are not. They are not subset of T cells, and they do not rearrange antigen-specific receptors like B cells and T cells do. They are experts on antiviral immunity, and what I described is called missing self-recognition. And their immunophenotype, they do not have CD3 on the surface because they don't have T cell receptor, but they have CD56, CD16 in many of them, CD2, and so they share a lot of antigens with T cells. But they should not have surface CD3 and no T cell receptor. And NK cell activity is regulated by a fine balance between activating receptors and inhibitory receptors. And without going into details, I just want to say that the list of those receptors is now a lot longer than this. And the idea is that the inhibitory receptors dominate over the activating ones to ensure safety. And I knew all of that at the time. That was known at the time, and I wanted to learn even more about this, and I ran in a wise book that United States is absolutely the best place to do this, and that was about 2005, when the green line is the research funding trend in United States funded by NIH up to 2005. So this is the time I came to do research that obviously nobody could have predicted, but it would be unreasonable to expect it will keep rising at this pace. And what we did is in vitro culture of hematopoietic precursor cells. So starting with very few of them, over the course of 2 weeks, we get a lot of cells already. From 500 cells, we would get 2 million cells. And then eventually, over the next 2 weeks, they would become natural killer cells. So that was a nice study system. It required stromal cells, which were called ELO8, and they were derived from mouse, as well as cytokines. And these are flow cytometry dot plots that show over the course of the culture, day 11, 15, 21, 29, 36. Those red dots represent natural killer cells. So you can see gradual emergence of natural killer cells and gradual disappearance, if you wish to see it that way, of those other larger cells. And NK cells derived in those cultures, they express pretty much all of the tested NK cell receptors that I showed in the earlier slide. They expressed activating receptors, inhibitory receptors, even some of the key receptors.
Bartosz, in the culture situation, what cytokines did you use to induce those?
So we used the most important is interleukin 15.
Yeah.
That's the critical one for NK cell differentiation. I would say the rest is sort of to stimulate overall growth of all the precursors that eventually IL-15 drives. So the other ones, we use stem cell factor and FLT3 ligand, which are rather basic at the early stage, but the main, and interleukin 7 as an overall lymphoid stimulator. But the key is interleukin 15. So what we observed is that not all NK cells expressed those receptors. There was always a subset that didn't. And there was an easy way to distinguish between the two subsets based on CD117, which is usually in other lineages marker of immaturity. It's a receptor for stem cell factor. And especially if we combine CD117 with CD94, we got a very nice split between two subsets, and we wanted to understand further what is the relationship between them. So it turned out that it's only the CD94 positive, CD117 negative subset that is expressing inhibitory receptors, importantly CD94, NKG2A, which is its partner actually in expression, and key receptors in some of them. And they expressed also the activating receptors, whereas the other subset did not express activating except for NKP44 and CD161. And didn't have inhibitor receptors. We also looked at the ability to kill other cells, and this is just to show that the CD117 dim subset are the cells that could kill because they express perforin and granzyme. And the same was shown in functional studies. So it seems like the 117 high, 94 negative subset is sort of less developed compared to the CD117 low 94 positive subset, which has all of the markers that we know that we've tested for NK cells. So natural suggestion was that maybe this is an immature stage of NK cell maturation, and this is how we published this. And we also did cytospins to look at these cells. I didn't make much of this at that time. Didn't know this is, for the most part, what I'll be doing now. So they have this interesting chromatin pattern, which is almost similar to erythroid precursors. It's really dense, but they are lymphoid cells. And the 117 low subset, 94 positive subset has cytotoxic granules, whereas this one didn't. So at the same time, Aaron Freud, working at Ohio State University, has described exactly the same things, but he was looking at secondary lymphoid tissues, tonsils for the most part, and so this is essentially what is now considered progression from stage 3 to stage 4 of natural killer cell maturation. At that time, I decided to pursue pathology residency, and while I was in residency, a new cell type has emerged that we didn't know existed at the time. And they are called innate lymphoid cells. although that name has evolved through several other variants. And what has turned out in the Mike Werner's lab after I left, that this subset, 11794 negative, contains a lot of those cells. So it's not just a stage of NK cell maturation, but it's also a diversion point to a different cell type. And we saw that because when we tried to differentiate 117 high into low, some of them did, some of them never did that. And there was a little bit of movement in the opposite direction as well. So in the schema of NK cell maturation, this would be the deviation point to innate lymphoid cells. This particular subset is called ILC3. We also did, and this paper actually gets a lot of citations, we showed that the same culture conditions can be used to develop NK cells from embryonic stem cells. That was together with the laboratory of Dan Hoffman and Peter Voll. And the interesting part was that these embryonic stem cells were a lot better at becoming NK cells, which at the time we thought, well, they're better at becoming NK cells. But now I look at it as the inability of these cells to actually become innate lymphoid cells, which has been since then shown that they are not, it takes a lot to make them become innate lymphoid cells. So in the schema of simple schema of hematopoietic development, NK cells are believed to be derived from common lymphoid precursors, which splits very early from cells that can become myeloid. And that's another view on the general idea of hematopoietic and other cell differentiation, is that cells have different choices, but as they make those direction, they choose a direction, it's very hard them to switch to another one. So that's the classical picture from a book from 1957 that people frequently show. It makes me think a lot of skiing, of a trail map on a ski resort, and there are different ridges and there are different places you can ski from, and you can start here and then over there. So this is a picture we looked at before. So this is the emergence of NK cells. And as it was explained to me, the idea is that all those large cells, the black dots, they just go away. They vanish because they're myeloid, and the NK cells emerge from the dust. But that's not really what we saw in those cultures. What we saw is that those myeloid cells are actually becoming NK cells, looking carefully day after day at different markers. So when we take very primitive hematopoietic precursors, they could not become NK cells with just the cytokines. It wouldn't happen. It required the stromal cells. But in the presence of the stromal cells, especially with hydrocortisone, which, if you think of the analogy with skiing, it's like we're grooming that trail for them. They do become NK cells. And they co-express CD13 and CD33 frequently when they become, which to me suggests that there's something else going on. So we did many different types of experiments. I'm just showing one. We chose MCSF receptor because cells that have that receptor respond to macrophage colony-stimulating, monocyte colony-stimulating factor, and that's the proof that they can and they would become monocytes. So we have sorted CD115 MCSF receptor positive precursors, and we have shown that they can become NK cells, and moreover, the NK cells derived from there are qualitatively different than NK cells derived from the MCSF negative ones. Moreover, this is just sort of to troll the system, to show that it's real. We added MCSF gradually to as it was becoming, as it was happening, and we could show even co-expression of CD14 at low level on those developing NK cells to prove that these are really the same cells. They're responding to both types of signals. So what we have shown, based on compared to the prior scheme of maturation, is that cells at different stages of myeloid maturation can become NK cells. So this is sort of, when we groom the trail for them, they go from one ridge to another. And since that time, the map of hematopoiesis has been heavily revised, and what we know now is that there is a subset of lymphomyeloid precursors which can give rise to both lymphoid and myeloid, granulocytic and monocytic cells. They don't really have megakaryocytic or erythroid potential. So this is now very well accepted that this is actually a very, very frequent way cells differentiate. And on that topic, what we do every day is we look at flow cytometry and bone marrow samples from patients. And frequently nowadays, patients are after therapy targeting CD19, which is marker of B cells. And here's an example of a 46-year-old man who had the diagnosis of diffuse large B-cell lymphoma and was heavily involved before treatment and then had quick relapse after initial chemotherapy, and therefore was treated with other modalities. And part of those other modalities was treatment with CD19 targeting antibody. So this was, in this particular patient, not CAR T cells, but the antibody was conjugated to a toxin. And he was referred to our center for autologous bone marrow transplant. What we found is an unusual population of cells which resided in CD45 dim side scatter lobe area, which for all intents and purposes looked like B-cell precursors, but they did not express CD19. At the same time, there was no evidence of lymphoma, and there was no evidence for other abnormalities, just by flow cytometry. And it was present at 1, 3, and 6 months after transplant. So the cells expressed CD10, CD22. They didn't express CD19 for the most part, but they did express CD10 at the level that B-cell precursors do. And they even showed some stages of stage 1 and stage 2 of B-cell precursor maturation. Didn't really become mature B cells. Why is this important? It's important because B cell leukemias and lymphomas lose CD19 to escape therapy. Now, these are not abnormal B cells. These are what we think normal B cell precursors. So, can they do that? Can they do something similar? Is there a mechanism for it? And the mechanism that it happens, that it occurs in leukemias and lymphomas have been well studied, among others, the laboratory of Dr. Andrei Tikhonenko at UPenn, where they showed that mutations cause changes in the splicing of the gene such that the expressed protein is lacking the parts that the antibody or the CAR T cells are targeting. So we have reached out to them. First, we needed to sort the cells, to purify the cells of interest. And the question was, did they lose CD19? And are they still benign? Are they abnormal in any other way? Or maybe do they correspond to a normal subset? The reason I'm showing my bike here because it was in February, and I biked over to the cancer center with a sample to sort those cells out. And in collaboration with our colleagues at UPenn, Dr. Asnani has sequenced the CD19 gene, and other than single nucleotide polymorphism, which was present in all fractions, the unsorted T cells, so it was not specific to this subset. There were no mutations in CD19 that would explain this. And also those alternative deleted RNA transcript of CD19 were not found. So this was not the explanation for what we saw. Meanwhile, a few months later, the patient came for another routine bone marrow biopsy, and at this time, he had perfectly normal B-cell precursors with CD19, with normal maturation, progression to a complete B cells with expression. So the problem took care of itself. It was transient.
Does the antibody stay bound to the...
We looked at that, and I did staining by IHC on the core biopsy, and it's also negative, which in conversation with Seattle Genetics, they said that would exclude it.
So I know with daratumumab, we see it stays bound.
Right, but for 9 months?
It's usually 4 to 6 months.
That's amazing.
So this is not the end of the story, though. Because we were thinking, okay, so that means the cells were okay. That was just a transient thing, and maybe they represented some unusual subset of precursors. However, in subsequent bone marrow biopsy, the patient has shown very small fraction of cells with monosomy 7 and now is fully developed therapy-related myeloid neoplasm and myelodysplastic syndrome. And we don't know, are we looking at a hat or are we looking at a snake who ate an elephant? This is from Little Prince.
What was the time course of the development of the MBF?
So first it was just cytogenetic finding without any morphology correlate, I think, in 1.5 years after transplant, and less than a year later it's full blown. And there's p53 mutation, actually 2 of them. Something we're thinking to test in those B-cell precursors. But this has created several questions. So going back to our idea of T-cell of cells differentiating, sort of like a skier that goes down the hill. What happens if we block one pathway, the one that they would most frequently choose? And perhaps that's what happens, that common lymphoid precursors, which vast majority of them, as we know it, go into B-cell maturation. Once it's blocked, they think of other options. They don't just die. And that may be a reflection of what we're seeing. Switching gears, speaking of immunotherapy, this is another study where we looked at immunotherapy of acute myeloid leukemia, and the University of Minnesota has probably the longest history of that, more than 10 years of experience. Many patients that were treated. And together with Dr. Curville and Laura Munch, who was earlier a fellow, we have looked at and retrospectively analyzed bone marrow biopsies, which were routinely obtained 2 weeks after the therapy. All patients included in those trials had highly refractory disease. So what we did is we stained the bone marrow biopsies for CD56, and it's not a perfect marker, but that's the one we have available at this time. And we compared the data and we counted the NK cells per square area. We compared the data with flow cytometry, and we also you had to have a control group, and that was a group of patients who received chemotherapy but did not receive NK cell infusion. And this is an example of patients who showed high degree of NK cells, high numbers of NK cells in the bone marrow in this early 2-week time point. So all those brown dots are NK cells. This is a focus of NK cells near a vessel. So we looked whether the presence of NK cells correlates with the clearance of leukemia, and indeed, patients who had increased density had more frequently, were more likely to have clinical remission, and patients, in looking at it the other way around, patients who responded had on average a higher number of NK cells per area in the bone marrow.
Bartosz, are those autologous NK or allogeneic?
Allogeneic.
Allogeneic.
And over the course of over 10 years, there were different clinical trials that differed with tiny details, but gradually the therapies have evolved. And we noticed that within those trials, there were actually differences. There were 2 that were standing out in terms of how many cases there were in the bone marrow. So we looked deeper. What are the tiny differences that make it? And we found that the use of Ontak, which is an antibody against CD25 combined with a toxin that eliminates regulatory T cells. So the addition of this antibody against CD25 resulted in increased number of NK cells, and also the use of total body irradiation, which was low level. It was not high level. Total body irradiation also resulted in increased engraftment and persistence of NK cells in the bone marrow. That was sort of new. The beneficial effect of Ontak was known. The problem is the drug vanished from the market for unknown reason, really. But the TBI is sort of a new observation. So in summary, we showed that NK cell homing and persistence in the bone marrow correlates with control of acute leukemia, and there are clinical factors which promoted homing and persistence of NK cells to the bone marrow, including the CD25 immunotoxin, working likely by depleting regulatory T cells, and also irradiation, and the mechanism here is not really known, but there are some animal studies that show similar effect. And the question is, how can we further improve homing of NK cells to the bone marrow and persistence? And secondary question is, how does the stroma, which we know in the bone marrow is abnormal in patients with AML, how does that affect it? So going back to the stages of NK cell maturation, I would like to talk about neoplastic diseases of NK cells now. Thinking of the maturation, the most immature stages, if they give rise to neoplasm, they present as acute leukemia. And the issue is that we very rarely call them as such because the current classification, the classifications have been evolving, but it's not easy to call leukemia acute NK cell leukemia. We actually had a presentation by Dr. Stone yesterday of a case which, if you think of the idea, what is the closest normal counterpart, that would be NK cells. But we still couldn't call it NK cell leukemia. It ended up called acute undifferentiated leukemia. Whereas neoplasm derived from mature NK cells include chronic NK cell lymphocytosis, which is sort of the NK cell counterpart of the LGL, large granular lymphocyte type of leukemia. It's a chronic but not very aggressive disease. And then there is a group of aggressive NK cell diseases presenting either as leukemia lymphoma or more as a lymphoma of the nasal tap, and they are almost uniformly positive for Epstein-Barr virus. So here's an example, and this is very similar to the case that we had actually in December, but it's from my prior training. Acute leukemia expressing CD56, CD7, some degree of myeloid markers, but not bright. And this was also called acute undifferentiated leukemia, but the thinking is that if you want to think of the closest normal counterpart, that would be NK cells. Extranodal NK T-cell lymphoma nasal type. It's predominantly extranodal lymphoma, as the name would suggest, and I really like the old name for it, which is lethal midline granuloma. And it tells the story, it's a very bad disease. And most cases appear to be genuine NK cells, but they can also have a T-cell phenotype. There is a genetic and regional predisposition in Southeast Asia, and the reason for that is not very well known. They have expression of markers of T cells, CD56, CD2. They have cytotoxic molecules, granzyme, and they are almost uniformly positive for EBV. And the characteristic features of this is they tend to affect vessels, and they cause a lot of necrosis in the tissue. They can also be associated with hemophagocytosis. They're almost uniformly positive for Epstein-Barr virus, and they express normal NK cell markers. This is one example of them. So we took up together with Dr. Balfour and Mike Verneris at the time the problem, which has still no answer really. How does the EBV get into NK cells? So we know what are the entry receptors for EBV in B cells. This is CD21, which is interacting with GP350. And also, there are also some co-receptors in that entry of EBV into B cells, and that's fairly well studied. And but do NK cells have those receptors? And the preliminary data showed that they do not. And in order to study this topic, we took advantage of special EBV virus which has green fluorescent protein in it. So once it gets into NK cells, the NK cells turn GFP positive. So as I said, NK cells do not express CD21, and they also do not express another receptor which has been recently associated with EBV. It's neuropilin. They're also negative. And when we cultured expanded NK cells and tried to infect them with this EBV virus, almost no cells became positive for green fluorescent protein. It's almost zero. So here is what the data looks like. Let me guide you a little bit through. So this is a B cell line which we use as a positive control without the virus, and this is B cell line with the virus. And with this dose of the virus, about 40% of them become strongly positive. It's another B cell line, which we used 2 B cell lines just to be sure, and this one was not that sensitive, and we'll get back to it later. NK cells, pretty much nothing, and we tried the same dose and double the dose and quadruple the dose of the virus, and still not a big difference, almost none. And we tried that in 2 different experiments with many different donors, and it's just...
Why did you look at cord blood versus peripheral blood mononuclear cells?
We didn't use cord blood in these particular experiments because we didn't have them.
Well, it just said UCB, so that's what I...
Oh, it's a symbol. It's a symbol of where the NK cells were derived, I believe. But that was, I don't think they were cord blood NK cells. They were, most importantly, they were not freshly isolated NK cells. And I'll get back to that later. We needed the numbers of cells, so we had to expand them in culture, and they went through special culture to just get decent numbers. So this is what it looks like with the B cell lines. A lot of them become GFP positive. But there was this other set of B cell lines that did not do it. And we didn't know why that happens. I actually talked to Dr. Mark Birkenbach at the time, and he was aware of that, but he gave me a different explanation than what we found. What we found is that they just don't have CD21 on the surface. The cell lines that are negative, they shed, for some reason, in some mechanisms, have lost CD21. And that explained very well the resistance of these B cell lines to infection with this GFP virus. So as we were explaining this project to multiple candidates, and I learned that hiring candidates is really a big task, and it's important, and and as we were going through the ideas together with Mike Verneris, we developed this concept that maybe what happens is NK cells interacting with B cells move away with a piece of B cell membrane, and that mechanism is called trogocytosis, coming from Greek word trogo, which means gnaw. And it's a process in which lymphocytes conjugate to other cells, and they extract surface molecules from these cells, and then they have them on their surface. So we hypothesized that maybe this is the mechanism that this happens. So if that were to be true, the co-incubation of NK cell with B cell lines might lead to acquisition of the CD21 and other B cell receptors. So we developed a plan to co-incubate NK cells with B cell lines that have and B cell lines that don't have CD21 and see what happens. So co-incubation with Raji caused a fair amount of NK cells to acquire CD21 on the surface, whereas co-incubation with B cell lines that don't have CD21 didn't. So we added EBV virus to these co-cultures, and what we saw is that co-cultures with B cell lines that do have CD21 resulted now in infection of NK cells, but not co-incubation with B cell lines that don't have CD21. So this is just a co-culture of NK cell, B cell, and the virus. We hypothesized that if we increase the contact between NK cells and B cells, that would happen more often. So addition of rituximab antibody, which is against CD20, indeed resulted in increased percentage of NK cells that were GFP positive. I didn't mention that, but patients who have defects of NK cell cytotoxicity are known to be a lot more susceptible to this problem, especially children that develop HLH. And we used concanamycin A, which inhibits acidification of cytotoxic granules and prevents perforin maturation, and therefore prevents cytotoxic granule release. So NK cells treated with CMA make conjugates with B cells, but they're unable to kill them, sort of mimicking the situation of inborn genetic defect. And indeed, treatment of NK cells with concanamycin did gradually increase the number of NK cells that were positive for GFP. Next, we asked the question, well, maybe the virus just attaches to these cells. We need to test if it's truly transcribed. So we used RNA staining. It's possible, actually, to stain RNA and detect it by flow cytometry. We used the probe specific for EBV. And it's not a high-level expression, but there were definitely NK cells which showed EBV RNA expression but it was nowhere near the level that we saw in controlled B cells. So in summary, NK cells lack known EBV entry receptors. The interaction of NK cells with B cells can lead to acquisition of CD21 by mechanism of trogocytosis. So this was not happening when we prevented contact of NK cell with B cells using transwell culture system. And such mechanisms can lead to EBV entry and viral gene transcription in NK cells based on this RNA staining. And rituximab increased the rate of the transfer and increased EBV infection. And inhibition of NK cell toxicity, mimicking perforin and other defects, increased the rate of NK cell infection by EBV. We showed, I didn't show data with transwell experiments, but NK cell B cell contact was required to see that. And we also found more recently that freshly isolated NK cells are generally a lot more susceptible than cultured NK cells. But if you put EBV virus on B cells, they become immortal. And they grow in culture forever without any cytokines. We never saw that with NK cells. So I'm not personally convinced that this is really the mechanism that it happens, that it's really sufficient to transform NK cells. We have just not seen that in our hands. Moreover, the study of this topic of EBV positive NK cell lymphoma is very challenging to us because this is a very rare disease in our practice. So here is a case of a patient who was otherwise healthy. He was so healthy that he ran an ultra marathon. Shortly before that, he presented to a doctor. He had liver symptoms, and liver biopsy showed hemophagocytosis in liver sinusoids. Bone marrow biopsy showed fairly subtle, but there was some hemophagocytosis also. And flow cytometry showed CD68 staining stains macrophages. It showed cells that are filled with other cells, mostly red cells. So this is the reflection of hemophagocytosis in both places. And could very well end at that, but we saw this type of cells in blood and in the bone marrow, and if you notice the quality of the chromatin, that is reminiscent of what we saw in those in vitro developmental cultures. And indeed, flow cytometry revealed a population of fairly large biforward scatter cells that were NK cells but had abnormal phenotype, loss of CD7, uniform expression of HLA-DR, and it was similar to what was described for for EBV positive aggressive NK cell leukemia. However, these cells were 56 positive, but they were negative for EBV. And it was not really clear what is the role of those NK cells in this patient's really clinically poor status. He was feeling very sick. And this was actually indeed aggressive NK cell leukemia lymphoma, but it was EBV negative. Karyotype was very abnormal in those cells. Over the course of next 2 months, he has gradually developed bone marrow necrosis and unfortunately passed away despite therapy. We did some liver pathology. Iron stain is done routinely on every case. So I looked at this and I noticed that there is iron granules. There are iron granules in those macrophages, and that made me think a lot about iron. First of all, there's the irony of NK cell research, and I felt like Don Quixote in this situation because here I'm studying EBV infection of NK cells and I get to diagnose a de novo case and it's aggressive NK cell leukemia, but it's negative for EBV. And I believe sometimes life tries to tell us something. And moreover, if you think of by analogy, there is a similar precedent with Burkitt lymphoma, which in the setting of endemic Burkitt lymphoma is uniformly EBV positive, whereas in non-endemic form, it's actually EBV negative, but otherwise it's essentially the same disease. So iron, I decided I need to do some basic study. I don't know much about iron anymore. And the further backward you can look, the farther forward you can see. So I learned, just opened the book and I saw this graph and it made me think actually, these are eons ago when there was no life on Earth. And photosynthesis started But life couldn't start until iron was quenched. There was a lot of iron in the oceans, and the active iron prevented development of life. So all life forms had to develop systems for handling iron because it's very dangerous in combination with oxygen. And I would like to learn what does it have to do with NK cells and hemophagocytosis. And because it's not known really how NK cells lead macrophages to eat other cells and develop this tissue necrosis. And I believe this has relevance not only for NKT cell lymphoma, it also has relevance for hemophagocytic lymphohistiocytosis, but it also has relevance to the rapidly developing field of immunotherapy. And this is just one picture of an example of a case which we saw cerebrospinal fluid of a patient who had B-cell lymphoblastic leukemia, was treated with CAR T cells. Actually, they were both CAR T cells. We think they were CAR T cells and the lymphoblast in that CSF. And we also saw this, a macrophage eating red cells and lymphocytes. And I don't think this is just pure coincidence. So in summary, actually I went pretty fast. NK cell differentiation, we talked about the NK cell differentiation pathways and developmental stages and the relationship with other lineages. We discussed a little bit the concept of lymphoid development being skewed in some way by the treatment preventing B-cell differentiation. For example, with CAR T cells, I showed the data which we have acquired studying NK cell immunotherapy and homing of NK cells and persistence of NK cells in the bone marrow. And it's an active topic of clinical investigation at the University of Minnesota with a focus of how can we improve it. And we talked about aggressive NK cell lymphoma and mechanism of NK cell infection by EBV in vitro studies. And then finally, I've initiated the question of why and how are NK cell lymphomas causing tissue necrosis and hemophagocytosis. And thank you for your attention. I have some people to acknowledge. Dr. Balfour Lab, who was very supportive in the EBV studies, and co-workers from the Cancer Center, my former PI, and Jeff Miller, Veronika Bakanova, and colleagues from the Department of Hematopathology, and Dr. Munch and Curville, who are now elsewhere, and collaborators from UPenn. And these are our son. He's showing his wingspan, and I would be glad to take any questions.
Thank you, Bartek. That was a really great talk. I have one quick question. So there have been reports of, for people that do a lot of B-ALL MRD, sometimes they find NK cells that aberrantly express CD19, or maybe it's a normal derivative, and I'm just wondering if you've ever seen that and what it might mean.
That's an interesting topic, and we looked at it together with Frank Cichocki from Jeff Miller's group, and he has done RNA expression studies using RNA-seq. And you can find expression of B-cell genes in some NK cells, and it's not all of them, but we don't really know what that means. But it seems like, yes, this could be a trace of those different developmental pathways that NK cells are taking.
That's a very nice and interesting talk. I'm interested in the dichotomy of the NK cells with the 117, 117 high and low. Because it looks like in almost every immune cells, it's becoming the same concept. So it started with T cells, then we got the Tregs, macrophages, now you have two types and everything. So it's interesting to know. So my first question is, the 117 high NK cells, you think they're the immature form. So what happens to them? Do they mature and acquire granzyme?
So yes, so we don't really see a lot of those very early cells. You would find them in lymphoid tissues, like Arne Freud did that. And we were asked by reviewers to prove that they are real, so we isolated them from cord blood. There were very few, but they were there, and they had similar properties, and they become the next stage. But this is stage 3 is really immature. Stage 4 corresponds to a fairly abundant subset of NK cells, which are called CD56 bright NK cells, and they represent about 10% of NK cells in peripheral blood. And then the CD56 dim NK cells is the fully mature NK cell population in blood, at least, and the subset that corresponds to them is probably the ones that express Q receptors that we see. But the definitive answer, I don't believe we have definitive answer. Is there only one pathway of NK cell differentiation or two? And we wrote a chapter, and recently there was a review. There are many reasons to think that there may be actually two different pathways, and some NK cells may get to the CD56 dim stage without going through the 56 bright. But that's still open.
So, I'm sorry, the second part of that question is, so you didn't show the flow data with the granzyme B, you know, with the immature NK cells, so I'm just curious.
So the stage 3 ones didn't have granzyme, stage 4 have less granzyme than stage 5. So there is progression in terms of acquisition and also ability to kill.
Bartosz, I'd like to go back to the last point on your summary slide and ask you to expand a little bit on hemophagocytosis. I've never particularly understood why people get hemophagocytosis. What's your current understanding why that occurs?
I have a theory, but I would have to prove it.
Okay, what's your theory?
So what is required, for example, for this is there is this mouse model which is interferon gamma deficient and it doesn't happen. And there is a JXMed paper from the same group from Cincinnati Children's that interferon gamma is required. But it's not really known from that point on what does it actually do to macrophages. But my hypothesis is that it has to do with iron. And there is a new form of cell death called ferroptosis. And I believe what it induces in macrophages is ferroptosis. Ferroptosis increases ability of macrophages to phagocytose red cells. In turn, phagocytosing red cells in combination with inflammation increases ferroptosis, and it's a vicious circle. I shouldn't have said that, but that's the theory.
Gotcha.
Have you anyone tried to infect EBV into the patient NK cells with EBV mother?
I did think of that, yes, because there is a susceptibility that's associated. We don't really know with what. It could be HLA, it could be some other polymorphism, but we didn't really have the opportunity to pursue that. Yeah, that was one of the plans that we made.
Great. Well, thank you.
Thank you.